Thanks so much. Ooh, thanks so much for the really kind introduction. I've set myself a timer, so hopefully I will not talk your ear off. But yeah, thanks so much for welcoming me here, and thanks to the ACAP folks for inviting me. This is a really important emerging issue. I just got off a call because there's even new information emerging every day, and it's something that's really kept us on our toes. And so what I'm hoping to do here today is sort of break down some of the important concepts about avian influenza. There's a lot of jargon. The situation has been emerging really quickly. And so I'm hoping to just break down those concepts, why they're important, and in a way that hopefully you folks can apply to the species and in the context in which you work, because I know it's not a one-size-fits-all type of pathogen, and in terms of mitigation and response, it's not a one-size-fits-all. And so that's sort of my goal here today. And I hear there's a club, and I'm a little bit worried about getting kicked out of it, but I'm actually not a seabird expert, so I'm going to put that out there right now. I do work with seabirds, and this is an emerging seabird story. But I also work with a lot of other wild birds as well in my role. And I'm going to talk a little bit more. Oh, here's my clicker here. Wrong way. I'm going to talk a little bit more about my role just briefly. So I do work for Environment and Climate Change Canada. It's a branch of the federal government in Canada, and it actually has quite a broad mandate. So generally the preservation of our natural world. And we do things related to pollution, to weather, to climate. But then the part of the agency that I work for is dedicated to the conservation, protection, and management of wildlife and their habitats. And specifically, our mandate is migratory birds and species at risk. Canada's actually had an interagency surveillance program for avian influenza and wild birds since 2005. Funding and interest and capacity in the program has varied over the years, as with most things wildlife related. I'm sure you folks can relate to that. But in 2021, early 2021, we really recognized the need to scale up our efforts and sort of change the way and adapt the way we were doing things, just given the context that was emerging in Europe. We rewrote our implementation plan, and it's actually available as a supplementary document to the preprint at that QR code there if you're interested. But this surveillance program is the result of many, many interagency partners in Canada. And so throughout the talk, I'm going to be peppering in some data from Canada. And so anytime you see that data, it's really only possible because of the work of these many, many partners. So I wanted to start out just by acknowledging them. Okay, so let's dive in. A goose Guangdong 196 lineage H5N1 HPAIV bearing the HA gene of clade 2.3.4.4b. Super sensical, right? It makes total sense. And before you get mad at me, I didn't come up with it. But what I'm going to be doing over the next several slides is kind of breaking down what this means. People use different components of it in different contexts, but this is sort of the full name of the viruses that we're dealing with today and that we're so worried about today. So we're going to start with the A. And what this means is influenza A viruses. So there's actually 4 different influenza viruses. They all fall under the same family. They're genetically somewhat similar, but they differ in particular with respect to their host range. So, for example, influenza B, C, and D are primarily mammalian viruses. And a particular example of this is influenza B, which is primarily a human virus, and it often causes seasonal flu in humans. But influenza A really stands out here, and hopefully that's a little bit obvious based on this figure here, because it has the widest host range. So, people get particularly worried about influenza A viruses because they can make these host jumps. But primarily, most of these viruses are adapted to avian species. Influenza A viruses are also known for their incredible diversity and their ability to change and diversify quickly. So this graph is starting in fall 2021 when we had our first detection of virus in Canada over the ensuing, I believe it ends in March of 2023. And you can just see the incredible diversity in how quickly the virus changes over time. So this is the number of genotypes that we detected in Canadian wild birds. There are two things that I really take from this. The first is that if we're not doing surveillance continuously, we're pretty quickly going to miss what's happening, right? This virus is changing constantly. The other thing is that not all of these mutations are leading to a gain in fitness in the virus. Some of them are neutral, some of them are detrimental to the virus, but some of them cause increased fitness. And those are the ones that we particularly worry about. And what that can mean is they can adapt to new hosts, they can become more transmissible, they can become more deadly. And there are two main ways that these viruses change over time. So they're highly transmissible, and so they can transmit very quickly from individual to individual. And these are RNA viruses, and so they're actually not very careful when they replicate. And so it's quite easy for them to get these point mutations introduced. And so every time it transmits, that's an opportunity for a new mutation to be introduced into the virus. We also get these contexts where we have new species that are exposed. And every time a new species is exposed, that's also an opportunity for the virus to adapt to that new species. We can also get what's called reassortment. And so what that means is if I'm a bird and my neighbor on the left has influenza, my neighbor on the right has influenza, and I'm unlucky enough that they both infect me at the same time with slightly different viruses, that genetic material can mix up and result in a completely new virus. So this is a way that the virus can change really quickly. And often we can be caught a little bit off guard with just how rapidly these viruses can change. And we're even seeing that now as news continues to emerge. And I'll show a couple of those headlines as we go through. Pigs. Let me just talk about pigs for a minute. So you may or may not have heard, but pigs are often referred to as mixing vessels. And so pigs were in the middle of that image on the first slide. And so they're actually one of the species that all of the influenza viruses can infect. And so often people worry particularly about pigs when they're at the interface with wild birds in particular, just because they can act as that mixing vessel for a bunch of different kinds of influenza viruses. And so multi-species interfaces can really drive genetic change. And so this is an example photo. I actually borrowed this photo from a colleague in the Northeast US because I think it demonstrates this really well. So this is a dead marine mammal being scavenged by seabirds with a coyote off in the distance. So who came in later to also scavenge on that carcass. And so this is an interface between marine, terrestrial mammals, avian and mammalian species. And so this is one of those concepts that you can hopefully take back to your own species and your own islands, what interfaces are present, and that can sort of factor into any risk assessments you might be doing in terms of virus introduction and virus change in those environments. Okay, so moving on to the H5N1 component of this really long, complicated name. So viruses have structural proteins and surface proteins, but the surface proteins in particular are really important for that virus to be able to bind to the host that it's going to infect. And so these are actually the regions of the virus that are most variable. And so we use these proteins to sort of further classify viruses. So when we say H5N1, we made subtype 5 of the HA or hemagglutinin protein and subtype 1 of the NA or neuraminidase protein. And there's actually 144 subtypes that have been detected. In this matrix here, the ones that are green are the ones that have been detected in wild birds primarily. And this is where we get terms like avian influenza and bird flu. These are the influenza A viruses that are most adapted to wild birds. The ones in yellow, H1N1 might sound familiar, for example, are the ones that are most adapted to mammals. And so H1N1 in 2009, you may or may not remember, but it caused a pandemic, and that was actually the result of mixing in a pig of avian, human, and swine influenza. Okay, moving on to these other two components. So on the left-hand side here, we have Goose Guangdong 196 lineage, and that's basically helping us to track the virus back to its original lineage. And so what this means is that the original host was a goose. In this case, it was a domestic goose in Guangdong, China in 1996. And this helps us track things back so we can understand how the virus has moved and changed over time. On the right-hand side, we have clade 2.3.4.4b, and that brings us to the current state of whatever virus we're looking at. If we look at this on a phylogenetic tree, we can see our original Goose Guangdong virus there, and then over the course of 20 or so years, the virus has adapted over time to where we are today at our H5 clade 2.3.4.4b. And we often talk and think about this as one virus, but it's really not. It's a whole bunch of different viruses that are similar but different from each other because of those mutations and changes that are happening along the way. So each of these viruses can behave slightly different in hosts. Okay, HPAIV, highly pathogenic avian influenza virus. So influenza viruses are classified as either low pathogenic or high pathogenic. And this is actually based on what they do in domestic birds. It actually really isn't based on their impacts in wild birds at all. So low pathogenic avian influenza viruses in a domestic bird will primarily be pretty focal in terms of what tissues they impact. So it'll be respiratory and gastrointestinal. So you can see those tissues in pink there. When we talk about highly pathogenic avian influenza, these are viruses that tend to be able to infect everywhere in the body, every tissue. And usually what this means in domestic birds is that they get very sick and they die very quickly, often. But when we sequence the virus, we can also tell the difference because there's the introduction of very specific amino acids in an important region of that genome. But what about in wild birds? What do we know about these two things in wild birds? Well, what we know is that low pathogenic avian influenza viruses have a long history of circulating in wild birds. Waterfowl in particular are known as a reservoir host, and what that means is that the virus can persist in those populations over time. And we know that infection is common in these wild birds, but disease is rare. So it doesn't often cause any clinical signs. We even see low pathogenic viruses circulating in domestic birds in some parts of the world. Highly pathogenic avian influenza, this is a little bit of my soapbox here, but wild birds are actually not a natural reservoir host. And I find that often in the media, wild birds are presented as a reservoir host of influenza viruses. And that's actually, it's a little more nuanced than that, right? So highly pathogenic avian influenza viruses are really a poultry virus. They came from poultry. And so what happens is low pathogenic avian influenza viruses circulating in our wild birds spill over into poultry and then undergo that mutation that we talked about, that change to highly pathogenic. And this interface between wild birds and poultry is a really important part of this story. And so in certain parts of the world, the interface between these species is really close. So you can have domestic birds grazing in wetlands even. So that's direct contact between these species. And what's happened is that there's been spill back of these high pathogenic viruses into wild birds. And originally, these wild birds were completely naive, right? And so they would immediately die. The virus wasn't able to sort of move beyond that in the wild bird population. But it had many opportunities to keep trying. And over time, it gained that function in order to be able to be maintained in wild birds. And this fundamentally is what is different about these 2.3.4.4b viruses. They are the first to be able to be maintained in wild birds and moved across large distances in wild birds. But then we also get crazy things happening like spill back and spill forth, and we still have our are low pathogenic viruses circulating in our wild birds, and so you can continue to get reassortment between those. And I do just want to go back for a second. So this X is really meant to show that often in our domestic birds, this is where we try to control this. So these often get depopulated in order to stop the transmission of these viruses in domestic birds. Much harder to control in wild birds. And certainly we would not want to be depopulating our wild birds. And that wouldn't be effective anyways. Okay. So what we know today is that when a wild bird is exposed to the current clade of highly pathogenic avian influenza viruses, we can have a couple of different outcomes. So the bird can be infected. Typically, we don't really see any clinical signs. We know a little bit less about whether there's sublethal impacts. We're starting to get a little bit of information about that there may be in some cases. In other cases, it looks like there's not. But we also get birds, of course, that are infected and die. And I realized when I was making this deck that I was a little bit biased towards my dabbling duck photos. And so we know it's not just dabbling ducks. It's many other species that also die. We know a little bit less about this middle category. So are there birds or mammals for that reason that get infected? They become clinical, but they live. We have specific instances in rehabilitation facilities where this has been reported, but we know really little about how often this really occurs in the wild. And in Canada, we do surveillance in both live birds and sick and dead birds. And what we can take away from these results in Canada is that the distribution of birds is actually quite different. And so in our live birds where we're detecting the virus, it's largely dabbling ducks. In our sick and dead birds, it's largely not dabbling ducks. So it's pretty much everything else. And we have to ask ourselves why that might be, right? And it's not an easy answer. It's a complicated question to answer, but we can think about the interaction between host, virus, and environmental variables, as well as how these interact with each other and accumulate with each other. And so if we go through a couple of examples, so dabbling ducks, so if we focus on the host circle for a minute, dabbling ducks we know have some degree of pre-existing immunity to low pathogenic viruses. They're the reservoir host. And so it's not the same virus, but it may give them a bit of a primer for when they're exposed to new viruses. We also know that young birds, just across the board, they're still developing their immune system, so they don't have that primed ability to respond. If we look sort of in the interaction between host and virus, what we know is that dabbling ducks co-evolved alongside this virus over that 20-year period, right? So this is a virus that's also learned how to adapt to the particular bird hosts that are at that poultry interface. But pre-existing immunity doesn't explain everything, right? Because we can still get a healthy dabbling duck that gets exposed to the virus and immediately dies. And so there are viruses that are simply just more deadly. There are viruses that transmit more easily. Hosts shed them more, so there's just more virus in the environment. So you're getting a higher dose when you're being exposed. And so part of the reason that seabirds may be more susceptible, I mean, it's clearly a combination of factors. There's not one answer, and it's going to differ between populations. But largely, seabirds have had less exposure to low pathogenic avian influenza viruses. They're largely not the ones that are interfacing with poultry. So these viruses haven't evolved alongside seabirds. They likely have less pre-existing immunity that might help them out. But also we have to think about what other stressors they have going on in their environment, because mounting an immune response requires energy and resources that they may be using to address other stressors at the same time. Okay, so let's go back to 1996. This section in blue is depicting low pathogenic viruses circulating in wild birds. In gray down here, we have low pathogenic viruses circulating in our domestic birds. In 1996, we have that change in domestic birds to highly pathogenic avian influenza. This was in Asia. We know it was in Guangdong in 1996. And then for a period of about 10 years or so, it primarily was a poultry issue. And so over this period of time, it was primarily moved around by the movement of infected birds and through the trade of infected birds. And so the number of poultry globally has dramatically increased over time. These are industries where the birds can be densely populated. They're often situated in key wild bird habitat, particularly waterfowl habitat. And so this is my second soapbox of the talk, if you will indulge me for a moment, is that I think for those of us that work with wild birds, we have a little bit of a role or a responsibility to communicate and to remind people that actually this is a poultry disease that is now causing widespread, dramatic, catastrophic impacts in some species of wild bird. And so thinking about how we can actually protect our wildlife from these livestock diseases. Moving on from 2005 and onwards, that's really when the wild bird story starts. And so this is when we start seeing those spill back into wild bird populations at the poultry wild bird interface. So thinking about what species are most likely to be present at those interfaces. And each of the sort of solid black lines there is demonstrating an important reemergence event. So either the virus moved to a new area, it moved to a new species, it fundamentally changed or had some sort of gain in fitness in wild birds. And so over this period of time, primarily how the virus is moving around now is wild birds and migratory routes. And so if we think about what species are actually causing this widespread movement, it's going to be those that are getting exposed and living. So birds and mammals that are getting exposed and dying are important for transmission, but on a more local level, right? So if we're thinking about these large-scale movements of this virus across the world, we're really focused on those species that are getting exposed and living. I do also want to just highlight, though, that wild birds are not the only things that can possibly move the virus, right? This actually is from yesterday. There was a human case of avian influenza that was detected in Victoria, Australia. I do just want to preface before we go any further that this is not 2.3.4.4b, so this is not the virus that's causing the panzootic today. But the reason I present this is just to demonstrate that humans can also move these viruses around, right? In addition to poultry. So poultry are still part of the story, but largely wild birds are what are moving these viruses around. Okay, so here we are in fall 2021 in Canada, pretty shortly after I started my job, in fact, we started seeing incursions of this Eurasian virus into Canada. And so again, what we talk about as one outbreak is actually the result of several different incursions of these highly pathogenic viruses from Eurasia, which was the epicenter of where these emergence events started. From here, this should hopefully look familiar from earlier in the talk. These highly pathogenic viruses then reassorted with the North American viruses that were already present, the low pathogenic viruses, in addition to accumulating those point mutations over time. And so we have a huge diversity of viruses that are now present that are part of this clade. If we align our migratory periods with these sort of increased rates of diversity, they align pretty closely with our wild bird migratory windows, which makes a bit of sense because there's more wild birds around at higher density. The virus is transmitting more, gaining those or having those opportunities to mutate. But there's also more low pathogenic avian influenza on the landscape to reassort with. So in Canada, there was a dominant reassortant virus that emerged in spring of 2022. And this is a phylogeographic reconstruction. So what that means is we're just tracing this virus and how it moved over time. You can't see any detections in Central or South America because we didn't have them at the time that this phylogeographic reconstruction was done. But you can see that this genotype dramatically expanded in terms of its geographic range. So it was a really fit virus that outcompeted a lot of the others. And at the time we did this analysis, 92% of the publicly available genomes from Mexico, Central, and South America were actually this genotype, or originated from this genotype. So what that means is that we had a virus that emerged in Canada and moved all the way south. So sorry about that, but again, I can't really take all the blame. But I put this here just to say that it's really important that we are communicating across borders, right? These viruses are not respecting borders, they're moving around. And so how can we communicate and share that information between ourselves? Okay, so now we're in a position where this virus is global in scale. It's really unprecedented in many ways in terms of the number of species, the geographic range, and it's continuously changing and surprising us. And so we really need to remain vigilant on this. And so the next few slides are, if you're not convinced already, or to hopefully convince you that this is unprecedented. So avian influenza has been around a long time. Big deal, like, why do we need to be worried? Well, it's no longer just a poultry and dabbling duck issue, right? Like on the wildlife side, it is an issue of conservation concern that's been recognized by many international bodies. The number of species, the impact on species, but also starting to think about what the cumulative impacts are. So those other stressors that these species are facing as well. So this is a diagram that just shows the difference in the number of species that were detected with this virus before 2020. So in blue, the bars that are at the bottom of the figure there, and then after 2021 in orange at the top. And you can see that the number of species has more than doubled. So this virus has gained the ability to infect a much broader range of wild bird hosts. Folks are really interested in albatross petrel, so I have highlighted those here. You can see that prior to 2020, there were a very small number of species that had detections. Since 2021, that has dramatically increased. And the impact on species. So this is data from Canada. Our three primary species that likely suffered population level impacts after the incursion of these viruses. I'm only going to talk about northern gannet, just looking at my time here. But globally, mass mortality was detected at 75% of gannet breeding colonies. In Canada, we lost as much as 30% of our breeding population due to highly pathogenic avian influenza. And based on monitoring data that has been done since, these populations have not recovered to date. This image is from a gannet colony in Canada prior to the incursion and after the incursion, just to sort of demonstrate the dramatic die-off that was observed from folks who were monitoring that colony. But these impacts are not just in Canada, right? They are across the world, and this is from a recent publication, and I think it highlights that the number of wild bird mortality events is increasing every year and expanding. And what we know is that we're data deficient here, right? Like, not everybody is counting these birds, not everybody's reporting this, so this is just what we know. We also have an unprecedented number of mammalian detections and mammalian impacts. This is just a small number of the wild mammalian species that we've had detections in. We've also seen die-off. So in the northern hemisphere, where I am from, our mammalian die-offs have been in like the tens to hundreds. In the southern hemisphere is where we've seen more dramatic die-offs, which I know you folks are probably much more familiar with, but in the thousands plus. I know we're wildlife people, but thinking about our One Health triad here, this really is a One Health issue that we need all hands on deck on. So in the domestic and livestock space, the impacts to the poultry industry have been catastrophic, right? It's unsustainable, the number of birds that need to be destroyed after incursions of this virus. It's having impacts on trade, on our economy, on our food safety and security. But we've also seen new species impacted, right? Dairy most recently, which I can tell you, nobody was expecting, at least of those that I talked to, so this really caught people off guard. So we always need to be vigilant and thinking about what we're not thinking about, what we might be missing. But also, just as a note, domestic dogs and cats have been infected and have died from this virus. So for you folks who are out in the field, who may have pets at home, just something to keep in mind. Thinking about that biosecurity from when you leave the field and go back home to any pets you might have. But then the human side of things. So we know that there's zoonotic impact. So the virus can actually infect humans. This is not common, and the World Health Organization still considers this to be low risk, but we are seeing detections in humans. So actually today, there was a second detection of this particular dairy clade 2.3.4.4b virus in a human that worked with dairy. So now there's 2 instances in the US of this. So again, data from today, right? Like, this is actively emerging. This is not a static issue. And then we want to think about the wider environment and ecosystem, of course, right? So all of these carcasses, domestic and wild, how is that impacting our environment, right? Where are these carcasses going? How much environmental contamination is that resulting in? And then we have our PPE. Super important. We need to be using it, but that's also all landfill material, right? So we're generating all of this extra waste as well. Okay, so I'm going to move on just to walking through some other things to think about if you're potentially responding to a potential outbreak situation. And so it's not a one-size-fits-all. So nothing I'm saying is prescriptive or that I'm recommending. I'm basically just going to highlight concepts and things to think about. And then, you know, it's really up to folks on the ground who have that expertise with the species and in the context in which they work to think about what makes sense and what's going to be applicable. But of course, the first thing we need to think about is worker safety. We know that humans can get infected. And so we always want to be thinking about what measures we need to put in place. I'm not really going to talk much about this. There is a workshop tomorrow which will go over PPE, and so I'll leave that discussion for then. We want to think about how to reduce transmission and mitigate impacts, and we'll talk a little bit more about that. But then also, what data do we want to collect? Are we going to count the dead things? If we are, how are we going to do that? Where are we going to put that data? Are we swabbing things? Just dead? Alive? What other metadata are we collecting? And how are we going to integrate that information to inform our conservation programs? And then how are we going to communicate that, right? So with our seabird community, across borders, with our interagency partners, so with our poultry partners, with our human health partners, and with the public. I'm really going to talk about these 2 things in the next few slides. So what can tip us off in the field that avian influenza might be the cause of any clinical signs or mortality that we're seeing? So I do just want to give folks a trigger warning. I'm going to play these videos. Don't want to see what a clinical seabird looks like. Don't watch for the next few seconds. But primarily, the symptoms are neurological. So we have birds that are behaving abnormally, landing in abnormal places. They're having seizures. They're stumbling around. They have what people call a floppy neck, as you can see in that gannet. We can also see birds that have conjunctivitis. So their eyes are swollen. And we can have birds that are in respiratory distress. So they're gaping. We've also had a few detections or reports of iris color change and corneal changes in some species as well. But far and away, what's going to tip you off first is dead birds, and often multiple species in a small area. And usually these birds appear healthy, right? This isn't a virus that's only picking off the poor doing birds. This is a virus that can take out a healthy bird that has some pre-existing immunity. So often the birds we're seeing dead are in good condition. But the other thing I want to mention is that this can look similar to other diseases. And so a precautionary approach is really useful, assuming it is until you find out that it's not. And this really highlights the importance of testing to make sure that we know that it is avian influenza. And so how do we do that? Well, we can think about how the virus is being shed. In these birds that are subclinical, primarily it's orally and through the feces. We can also detect the virus on feathers in some cases. In our dead birds, the virus is likely everywhere, right? That's why it's causing these clinical signs, because the virus has spread throughout the body. And so if we want to test for the virus, we're going to think about what we're testing. So if we have a bunch of sick birds or mammals, we want to take the carcasses that are in the best condition. And if you can submit those to a lab, that's ideal. If you can't, I know you folks are working in remote areas, you can collect tissue samples or you can collect swabs. And I'm not going to speak to the specific protocols. There are protocols out there in order to do this safely and to maximize your ability to detect the virus through these samples. If we have live birds, we're going to want to take an oral swab and a cloacal swab and put those together in a vial for testing. We can also test sediment samples, so environmental samples, to detect the virus as well. And we're asking here, is there virus present? But what we know after infection is that the viral load in an individual will increase quickly. If the bird's going to die, the rest of this really isn't relevant. You're going to find virus throughout the body, usually in large numbers. But if the bird's going to live, it's going to amount an immune response, and that viral load is going to decrease. So we have a short window of time only where we can detect the virus. I'm not saying don't collect swabs because we're unlikely to detect the virus. The swabs are really important. We need that to get the sequence data to understand how the virus is changing. But what I am trying to highlight is that we have this other opportunity to understand what's going on by testing for antibody concentrations in birds, because antibodies last quite a bit longer. So we have a wider window of time to be able to detect exposure. And so what we're asking here is there evidence of past exposure and survival when we're testing for antibodies? So if you are collecting blood for other purposes, maybe you are already testing for avian influenza antibodies, but this is just to highlight that the gold standard sort of sample for this is serum. So if you're collecting whole blood and spinning it down, do you have serum sort of remaining from the other tests that you're doing to be able to submit for antibody testing? Depending on your lab, you can also put whole blood on an abuto strip, which is that filter paper up there, and they can get a result off of that. In Canada, what we have been doing is testing for antibodies in eggs. And so antibodies will be present in the yolk of the egg. And what that sample is going to tell us is it's a proxy for exposure in the female, right? In Canada, we have a national widespread egg collection program in place already for contaminants. And so we've basically hijacked that program to be able to test a broader range of species that are harder to kind of get your hands on for blood samples, to be able to understand exposure, because that can help us assess risk to those populations. So are they still around because they were exposed and they lived, or are they still around because the virus hasn't gotten there yet? Okay, and so thinking a little bit about how the virus transmits on sort of a more a finer scale, so we can think about how we can mitigate. And remember, these are options. They may not all apply to you. It is also an option to not respond or with a specific management action, right? So basically I say that just to say I'm not endorsing any of these methods. It's going to depend on your context and what your objectives are, but let's just walk through what some of those options are. And so we can have direct transmission, and what that means is physical contact. So one bird has physical contact with another, and the virus can transmit in that way. In Canada, we bait birds for banding, and so what that does is it artificially congregates birds together. And this is a density-dependent pathogen. So this means that physical contact and spread of this virus is going to increase in these situations. But we can also think about colonial nesting seabirds that are nesting in super high densities too, right? This applies in that context as well. We can also think about direct transmission through scavenging. And so a bird that's died is sort of teeming with virus. And the amount of dead birds and mammals on the landscape in some cases are sort of easy pickings for a scavenger or a predator. And that's going to be a whopping dose of virus that's being ingested. And so this is often why we're seeing these individuals die, and quite quickly. So if we think about how we can mitigate some of these direct transmission mechanisms in Canada, we thought a lot about whether we should stop baiting for banding, because what that's going to do is decrease artificial congregations of birds. We can also vaccinate wild birds, and they have done that in a very specific circumstance in the US, in California condors. For wild birds on a more broad scale, this is probably pretty infeasible. It's going to require booster doses. We simply don't handle the number of birds required to have that level of immunity. We can also remove carcasses from the environment. So what that can mean is actually physically removing them, taking them away and disposing of them, or simply burying them as well. And then we do need to think about direct transmission routes to ourselves. And so thinking about what PPE we want to use in the field in order to protect ourselves from this direct transmission. We also have indirect transmission. And so thinking about our fomites, so that just basically means an inanimate object that the virus can survive on for some period of time. So our boots, our trucks, our gloves, when we're in the field and interacting with these birds, can be a mechanism for the virus to be transmitted. We can also have environmental transmission. So the virus gets added into a wetland, it contaminates sort of standing water, and a bird is going to go and feed. And this is another reason dabbling ducks are so important in the ecology of this virus, is because of how they feed and the environmental contamination that we often see. So we know that the virus can survive in the environment for extended periods of time, and so that's why this mechanism of transmission is particularly important. It does depend on some environmental variables like temperature, humidity, etc., but something we should be thinking about in terms of how to mitigate this transmission. So again, we can remove carcasses from the environment to prevent them from contaminating the environment further. We can not go into the field. Or we can prevent the public from going to those sites if that's something that is allowed in that context. If we are going to go into the field, we can clean our gear, right? That's really important to make sure we're disinfecting any gear we're bringing in. Okay, so I'm going to wrap up there and really just leave you with some resource documents. So it can really help to be prepared to have an outbreak plan in place. Think about what data you're going to collect. Think about what PPE you're going to need. And these are all international resource documents that are publicly available that provide a lot of great information. If you are interested, in Canada we have developed a lot of protocols and policy and guidance which we have made publicly available as well. Of course, this is not prescriptive and it's not going to replace anything you already have in place, but you're welcome to review and take any pieces that might be useful. And I'm going to end there.
Thank you very much, Jolene. We have time for questions if you have some prepared.
Thank you so much. It was really interesting, really interesting. And just curious, you mentioned it could survive in sediments, and how long is that going to last for? Is this something that in a few decades we'll be seeing in lake sediments? And does that mean, for those of us who work with sediments, we could be looking back at relatively recent, virus or something pulling out past occurrences?
Yeah, that's a great question and something we're still untangling. So to address the first part, how long can it survive? This isn't my favorite answer, but it depends, which is the truth. And so it depends on a variety of factors like the temperature of the water, the salinity of the water, and various other things. But what we know is that in the optimal conditions, it can survive for extended periods of time. So they're recovering viruses from ice melts from like decades ago, right? But there's a difference between detecting virus and detecting live virus. And that's a bit of a nuance, but detecting viral fragments can tell us about what was there. Live virus means that it's actually dangerous and infectious virus. But yeah, we can, and we do currently use this technique for avian influenza, but they also use it for things like SARS-CoV-2, like wastewater surveillance for public health as well.
Hello, I was quite interested in the antibody screening, and I was wondering how much serum or whole blood you would need to generate the amount of serum to test those sorts of samples. And also the filter paper, do you need more blood on those or the same amount?
Yeah, great question. And so it's a little bit lab dependent and dependent on how many different tests you want to run. So there's more than one antibody test. We can test for whether there was exposure to like any influenza A virus. And then once that's positive, we want to dig a little deeper. Okay, was it an H5 virus? And so with each new test you run, you need a little bit more sample. But in general, we usually need 0.5 mils. That's sort of the gold standard recommended by our reference lab.
Serum.
Serum. Yeah, so we typically, like I work mostly with waterfowl and geese, and we're usually pulling about 2 mils of blood. I know on smaller species that's really not feasible, so that's why we moved to the Nabuto strip. And so what we're currently doing is trying to validate that to see if we can come up with a conversion to compare it with serum, so we can get those quantitative values off of that sample. But we are getting positive detections, so far the results are good.
So the strip's more just for positive negative screening, but you can't get that additional information?
We've been able to do the positive negative and the H5 screening with Nabuto, yeah.
You mentioned that the antibodies can be transferred to eggs and you can test the eggs. How likely is it that the virus itself is also transferred to the eggs?
Yeah, so we know vertical transmission does occur. We haven't detected any in our samples, and we actually don't know how commonly, but we do know birds have been exposed and go on to lay. It may just be that they're not being exposed or they're not actively shedding virus when they're laying. So in theory, it's possible. I haven't seen any detections in eggs, at least that I'm aware of.
Thank you, your talk's excellent. And it's a series of these talks that's occurring everywhere. So you say outbreaks commenced in Canada in 2020, it's now 2024. Year on year, how are the gannets going in terms of the reproductive success, in terms of, is this a repeating outbreak each season?
Yeah, so we were really worried about that. So we had our first incursions in fall of 2021. Our gannets started dying the following spring and summer 2022 in large numbers, and we were kind of bracing for 2023, and it didn't come. And so we're still trying to untangle whether that was because there was just less virus on the landscape, or whether there was some degree of population immunity in those individuals that did survive. We have seen some evidence of antibodies in some of our gannet colonies, and so there's probably a little bit of that going on, but we also just across the board in Canada saw fewer detections in 2023. It's not uncommon for viruses in general to kind of have this cyclical pattern where every few years they'll really ramp up in prevalence, and that's likely just due to this cycle of immunity that happens in some of these birds that are primarily driving it. So they're shorter lived species, so you know the population is naive, completely naive every few years or so. So yeah, going back to your original question about the gannets, we know that populations haven't recovered. We know that they are breeding and they are laying, the ones that are still around, but there is some variability among colonies. And there is a publication, I won't scroll all the way back because that'll make you dizzy, but there is a publication available where my colleagues who do this population level work have provided their current assessment of gannet populations in Canada. And so that's available as a preprint if you're interested in more information on that.
With regards to the gannets, I'm curious. So you mentioned that 75% of the colonies were affected, right? Do you have a sense if it was gannet to gannet transmission like spreading between colonies, or do you think that might have been several different introductions to each gannet colony?
Yeah, that's a great question. That's still being unpacked. There's been some suggestions that it was gannets that was moving it around, but actually probably what's more likely is, you know, maybe gulls or things that were visiting the colonies. I think the gannets themselves were so heavily affected, I really don't know how many of them did actually get exposed and survive. That's still something we're trying to unpack, but at this point in time, I wouldn't be comfortable saying that it's likely the gannets were moving it around. But there is also a preprint available that tries to untangle some of that as well. I can put that up on the slide after if you're interested.
We have time for one more question.
You mentioned that a vaccine is available to be used for California condors. Is the vaccine, like, is it available in general to anyone, or how would one go about getting a vaccine?
Yeah, so the California condors were vaccinated in the US, so that's actually outside of my jurisdiction. I don't have all of the details, but no, there isn't a vaccine that's widely available. We also don't have a vaccine that's been validated or known to be effective in wild birds. That's work that's ongoing, and we were really looking to the California condor kind of pilot study to see whether that was going to be feasible for some of our species that we were really concerned about. But when we're talking about a vaccine, there's a couple of different things, right? So there's a vaccine that can stop a bird from dying, but what we really actually want is a vaccine that can stop the bird from getting infected in the first place and shedding. And we don't have that either. Yeah, even in poultry, there's not, like, I know there's vaccine programs that happen elsewhere, but in Canada and the US, they're not widely available for poultry either.
Thank you very much. We're going to continue with our program, but it was a really interesting chat.
Thanks everybody.